Hello, listeners. Yamina here. Welcome to the DrGPCR podcast. In part one of this series with Celtarys Research, Dr. David Hodson took us through the physiology, how GPCR signaling shapes metabolic control, and why seeing receptors in real tissues is essential for answering the right scientific questions. Today, in part two of this podcast series, we shift to the chemistry that makes that visibility possible. You're about to meet Dr. Johannes Broichhagen, in short, JB, the medicinal chemist building fluorescent ligands that reveal where GPCRs live, move, and signal in live cells, intact islets, and even in deep brain circuits. His work turns abstract pharmacology into something you can actually watch in real time. As I mentioned, this episode is part of our ongoing strategic partnership with Celtarys Research. Celtarys Research develops and advances fluorescent ligand technologies so GPCR labs can generate clean, reproducible insights without relying on radioactive approaches. This series of podcast episodes are all about generating better GPCR insights, from physiology to chemical tools to the decision those insights drive. Terry's Corner sits on that same continuum. Dr. Terry Kanakin distills the GPCR pharmacology principles that help discovery teams interpret data with confidence and make mechanism-based calls faster. Link to Terry's Corner can be found in the show notes. And now, let's dive into this part two.
Hello, everyone. This is Yamina from DrGPCR, and today I'm very excited to have with me Dr. Johannes Broichhagen, and I did pronounce your name properly. JB, it's very nice to have you on the podcast. Before I ask you to introduce yourself, I just want to mention that this podcast is part of a 3-series episode that we're recording with our friends at Celtarys Research, and the next episode you'll know how the 3 podcast episodes come together. JB, welcome to the podcast.
Yeah, thanks for inviting me, Yamina. Very nice to be here.
I'm very excited to have you on today. Let's start with a little icebreaker. If you weren't a chemist, what would you be doing today?
Oh, no idea. Like when I was a child, I always wanted to be a soccer player, but I'm a very realistic person, and I think like in 5th or 6th grade, I figured out it's not going to happen. I thought about, after my A-levels, which were quite bad, to study philosophy. But I had a good mentor, like during my social service, who said, like, you need to do something where you can use your hands, like go with life sciences, and that was a good advice. So started with chemistry. But what could have happened? I don't know. I like aesthetics. I like working with my hands, as I said. So maybe a carpenter or an engineering drawer, something like that. But actually, I'm doing this since more than 20 years, so it never really crossed my mind again.
That is wonderful. That is wonderful to hear. Well, I think you're our first potential aspiring soccer player. Initially, we've had David in the last episode. He wanted to be a veterinarian, which he was our first veterinarian. We've had people who wanted to be doctors or everything but research scientists. We've had astronomers, but I think you're the first aspiring soccer player. Welcome to the podcast.
Thank you.
But let's start at the beginning. Would you please introduce yourself? Tell us, what do you do and where do you do it? And it's a given. You're a chemist. I think you're also our first chemist on the podcast.
Oh, great. Okay. Yeah, I'm Johannes Broichhagen, or in short, JB. I am a group leader at the Leibniz Forschungsinstitut für molekulare Pharmakologie im Forschungsverbund Berlin e.V., or in short, the FMP. It's a non-university institution. And we conduct research like in 3 sections, but mainly to address different diseases from a molecular standpoint of view. I studied chemistry. I started about 20 years ago, and then decided, without any background, I never had a lecture in biochemistry or molecular biology or anything like this. But then I was interested how life works. And like being a chemist is a very naive view on things, but I took the chance to go into chemical biology. So I thought, I still can do chemistry, what I learned, like organic synthesis, but can also maybe learn and apply the things I do in a biological setting. So this is where I'm coming from.
Wonderful. When did chemistry enter during your schooling, your studies? When did the chemistry enter your track?
I think 9th grade was the first chemistry class, and I had a very good teacher during the first years. So I got excited, but it also it gave me a lot of security. It was like one of the topics where I did not have to study and I was still okay at.
For me, interestingly, it was math, where I've always loved the logic behind solving equations.
Yeah.
And it had this soothing effect of I wasn't studying, but I love to do the exercises. I'm just wondering if you had the same feeling with chemistry at the time.
Similar. Like the periodic table always gave me security, right? Like, you know exactly where something has to be. You count through it 1, 2, 3, 4, 5, 6, 7, 8, and so on and so on. And also the history about it, like some elements were not known to a certain date, and then people found out about it and could place it together. So that always gave me security, like where I was somewhat in the realm of chemistry. You know?
That's wonderful. We'll get to the GPCR research and to the work that you do today, but I'd be curious to hear about what got you into research or how did you end up opening your own research group?
Yeah, it was during my studies. I had a couple of moments where, like, these aha moments, right? When you're sitting in a lecture and you're like, okay, now I got this concept. And I collected those and thought about like how you could engage with people and like teach them like concepts if they are more on the easy side or like more on the harder side, but like with metaphors. And this is what I wanted to do later on, like to stay in academia and then somewhat like share the enthusiasm and the fire I have for science with others. So then, of course, like I did the usual path, like doing my PhD in chemical biology, then went for a postdoc, became departmental group leader at the Max Planck Institute, and then finally independent here at FMP. And I was also teaching organic chemistry at Humboldt University in Berlin.
That is fantastic. And where do the GPCRs enter the realm of your lab and your research?
I was also during my PhD, so I was initially starting working on ion channels, so also transmembrane proteins. And this is how I got in contact also with David, who did the podcast before. And then in 2014, just before I graduated with my PhD, I went to a conference from the EASD, so the European Association for the Study of Diabetes in Vienna, because I wanted to hear a little bit more about like diabetes and so on to get ready for my defense. And there, like GLP-1R was like the really hot topic, and I got engaged in that. And then we first put this under optical control with small compounds, and then the journey went on and on and on GPCRs, starting with this to where we are to date.
And what got you interested? I mean, you mentioned GLP-1R. It's still very up to date. It's bigger than ever. But what got you interested in the field of diabetes and obesity? And you mentioned being in chemical biology, which I think it's really that gateway towards biology using chemistry and the chemical tools. But why specifically this field on the biology side?
So I got very excited, like literally and figuratively, because with ion channels, we were looking mostly at neuronal tissue. But then I realized beta cells and neurons, they have some things in common, right? They're geographically like quite far apart, like in the brain and in the pancreas, but they're both excitable. They share big parts of the exocytotic machinery. And then I also saw like, okay, there's this real demand and urgent need. It's a real world problem, diabetes. And this is what really got me interested into it, to contribute to a better understanding. I'm not an MD, so I'm not going to therefore healing or treatment, but like to look at it from a molecular standpoint of view. Like I, as a chemist, like look at carbon nitrogen bonds or whatever, right? And this is also like how these GPCRs are made up ultimately. So I thought I maybe I could help in that regard a bit.
And is this conference that you mentioned going to where you got into touch with David for the first time? I'd love to hear about how did you end up meeting him, but also collaborating together?
Right. No, this was before, actually. It was also during my PhD. I think David, as I know the story, approached my PhD advisor at a conference about the optical control of the ATP-gated potassium channel that is important for insulin secretion. And I was working on sulfonamides, and it is only one synthetic step to make sulfonylurea out of it, which actually blocks this channel and then leads to depolarization. So my PhD advisor wrote me an email, and I was at that time in Vienna doing a research stay, and asked me to make the molecules with David and CC. So I think my first email to David was like, "Dear Dr. Hodson, let me introduce myself. I will do the molecules once I'm back in Munich, where I did my PhD." I synthesized them, and we were in touch, and sent it to him. I think he recorded some preliminary data. And based on this, I secured funding for a short research stay in London, where David was at that time at Imperial. And then was there and did a lot of confocal microscopy And yeah, we got to know each other, of course, and then became friends and collaborators since 2012 it was. In London, I was 2013. I remember this, the Champions League final was there, Bayern against Dortmund.
It's so interesting as you were mentioning this. This is exactly that interface where the rubber hits the road, where the chemistry and the biology come together. Because any molecule, well, you can synthesize as many molecules as you want, but if you cannot test them and if you cannot see what these molecules do, they're useless. And you can do all the biology you want if you don't have the right tools to actually visualize and better understand the biology. And it's such a nice example of, cross collaboration.
Yeah, it's also what I adore, like this interdisciplinary of it.
I agree. When you mentioned doing some confocal microscopy, I'm curious to hear about the molecules that you synthesized at the time that you used while during your time in David's lab. Were those molecules used to develop tools and visualize proteins on the cells, or it was ligands that you were developing?
No, it was ligands we were developing, like photo switches, so to endow biological function with light. The imaging part was we did calcium imaging in the islets. So loading them and then looking at like oscillations and how we can turn these on and off, which ultimately lead to insulin secretion. That was the starting point, but it never had anything to do with GLP-1R, with any GPCR at that point, not specifically, but only like to gate hormone secretion.
Wonderful. What was your first impression when you actually were able to look at cells? Calcium activity in cells. Visually, it's such a pleasure to look just at cells.
So the first impression, I remember this, like when I was taught mammalian cell culture on HEK cells, right? I was like, wow, this stuff is living, right? Like what you have in the tube is living. Like in chemistry, you usually like have your organic solvent and a couple of compounds and a catalyst maybe. That was my first impression of it. And that is deeply rooted inside me. Like I still remember that quite well. And then, yeah, looking at activity, first, before I did fluorescence imaging, looked at, I never did it myself, but like patch clamp electrophysiology to derive currents from the cells. It's like, wow, yeah, you can look at this machinery somewhat physically. And then optically, it was like, yeah, you see these like flashing on and on, like these calcium waves. That was quite fascinating. And yeah, it still resonates within me. I still love doing that, I have to say.
Wonderful. And it's a beauty. And to be honest, even just culturing HEK cells, to your point, just looking at the cells under the microscope, I still find it fascinating. I have to admit that I had a hard time also splitting cells and throwing away whatever cells we were not using because, they're living cells.
Yeah. There's also a good anecdote, like sometimes when I'm sitting at our STAT microscope, and you acquire super resolution images, right, with these probes that we are developing, and you're looking at like small nanodomains or whatever, and then like I get excited so much that I like take my phone out and then make an image of the super resolution picture and send it, I don't know, via email to David, and it's like, okay, you get a really bad resolution out of it, but like you want to show it immediately, right, what you have acquired.
No, I agree. I know the feeling. I definitely know the feeling. Speaking of visualization, what was the first bit? Sorry, I will restart. What was the first big challenge you faced while developing the tools that you've been developing and working with David and his team?
Right. So, like initially, we did not think about visualizing GPCRs. Right? We wanted to control them one way or the other, pharmacologically, of course. And then I learned that good or validated antibodies are actually lacking against this important receptor. I thought, okay, that's actually, like from a chemical standpoint of view, a rather easy approach, right? I was taught in structure-guided design, so I had a look at back then there were no cryo-EM structures, but there were some crystal structures of the active domain bound to peptides. So I knew, like, okay, if we make a peptide, I know where I can attach a fluorophore, for instance. And then the challenge was I never made peptides myself. So, but I was then at the Max Planck for medical research with Kai Jonsen, and there was another postdoc who did his PhD mostly on peptides, and I teamed up with him, Tom Podevin. and asked him, like, hey, can you make this peptide and we'll conjugate it to a fluorophore? And that also shows you like the beauty of collaboration and how these things got developed. So like actually on the first paper, David, Tom, and I are corresponding authors, all of us.
Wonderful, wonderful. And from a visualization standpoint, I do understand that, initially you wanted to develop compounds or peptides that allowed you to modulate the receptors. But when does the visualization come into your collaboration with David, but into your work in general? And to your point, there isn't a lot of good GPCR antibodies that are reliable, and you obviously want them to the antibodies. You want them not to have an effect on the function, but just allow the labeling. I'd be curious to hear about, from a timeline perspective, when does that shift go where you finally got to a point where you have those molecules that are labeled and allow you to visualize receptors?
How that transitioned, you mean? Yeah, I was just, I don't know, fascinated is probably the wrong word, but like shocked maybe that like these receptors are so well characterized in terms of like their canonical functions, right? I mean, like what ligands engage, like what the pharmacology is, what second messengers are being generated, and so on, or like even atomic coordinates. But that we had a little understanding of like how this receptor behaves, like dynamically, like within the plasma lemma, for instance. Where is it actually, like in which tissue is it expressed, right? And to what extent? Can we quantify this to somehow? Yeah, these were all things where I said like, okay, we can maybe, and on top of that, can we have something that addresses the pharmacological relevant pool, which is the pool that sits on the cell surface and is surface exposed with respect to like receptors that are still trafficking or like an endosomal compartment are being degraded or being newly synthesized. So these were all considerations that went into the development of our Luxendins.
And what are some of the breakthrough moments that changed the trajectory of the development of these tools? Any difficulties, any stories behind the scenes? When one of the reasons why the podcast exists is to really go behind the scenes and tell the untold stories that you cannot put in the paper, to say, well, we've tried X number of times and then we had to tweak this list of things, and that's when it started working out.
It was surprisingly straightforward. I would say the breakthrough moment, like when we had the first images, I remember David was like calling, it's like, I can image through the whole islet, like which is 100 to 200 micrometer in diameter. And this was like a breakthrough. I said, like, okay, we got the technology working. Now what are we going to do with it, right? And so I was immediately like drawn towards, okay, we have to do super resolution imaging. And then other people were like, okay, we have to look at different expression sites and show that this is actually working. And others were saying like, whoa, you haven't proven anything specific yet. Maybe you start with this first. Then another collaborator was like interested in the dynamics. So all of this is in the first paper. And like behind the scenes, if you ask for this, is like we chatted with a lot of people, and they were all like excited. And I do not recall how many authors are on there, but actually a lot who contributed all like their little parts and showed the versatility of the probes themselves. And that was amazing.
It's so interesting. I've only worked one time on a project on CCR2 chemokine receptor where everything just went smoothly, where I had to run the experiment. I did my n = 3. There was no n = 4. There's no exclusion. Yeah, it was just perfect throughout all the experiments. So it's nice to hear, that the generation of these tools was smooth, and you were able to, again, going back to that cross-functional collaboration, to send them around and have people validate the tools that are now available to the general public.
Mm.
Why are the chemical tools? And it sounds, I've been debating about asking this question or not, because I, in a sense, know the answer to the question, but I want to get your perspective. And the podcast listeners are PhD students, postdocs, but also PIs. Why are some of the chemical tools more reliable than antibodies for GPCR studies?
Yeah, that's the ultimate question. I think like reliable should be taken with a grain of salt. I mean, both like chemical probes as well as antibodies both need careful validation. I think chemical tools have the advantage that they're easier to quality control, right? You don't have, or they're more reliable in their batch number, maybe, with respect to antibodies. They're usually more stable. And of course, what comes with it, they allow different experiments. Right? So antibodies are usually used in fixed tissue, and small molecular probes you can use in live cells to image these. You can also, as I mentioned before, you can design them in a way so you can discern protein pools, for instance. So you label the extracellular with one color and the intracellular with a second color. So this is what chemical biology offers to add to the portfolio. I would say like a very good, or like, yeah, a very good validated antibody totally has its merits. Yeah. It's always the question what you're looking for. Another thing is like from a super resolution standpoint of view is just the sheer size of it. Right? We address the orthosteric side, so the fluorophore is like within, I would say, a couple of nanometers from the receptor itself. But like if you would use a primary antibody, which is 150 kilodalton, and then you add a secondary antibody on it, so you're like 20, 30 nanometers far apart. And with the revolution of super resolution imaging and like the latest developments that came out, where you can actually like go sub 10 nanometers, right? That this really becomes important.
And I think the localization as well of your receptor, if you're trying to image in tissues, it's much easier with small molecules than with antibodies.
That as well, yes. That has another layer of complexity, of course, the three-dimensional tissue setting.
How do you balance creativity in tool design versus rigor for reproducibility?
I would be a little bit careful to say to balance this. I would say you can be very creative in your tool design, and when you make a bad tool, you'll reproduce bad data. So I would more say like be creative, and what I do is from the get-go, have a backup plan, or maybe like parallelize things, right? From a design point of view, and also like from a synthetic point of view. So what really drives data or experiments forward if you can do them side by side, right? If you have your 8 wells and you have your 8 compounds and you can test it on the same transfection, on the same day, same humidity, same moon, and whatsoever, right? Then to do it like stepwise by optimizing and going back and controlling for everything. I think this has sped up our approach quite a bit.
And I think you make an absolute great point. I've always been the proponent of parallelizing things, and the more you can test within the same conditions, sometimes especially with cells, star alignment is very important. Sure. But you never know with the cells, and it's more important. And so I'm curious to hear a little bit more about when you go ahead and think about designing new chemical probes, new tools against specific targets. Do you collaborate with different biology groups in order to get a sense of what is needed within that collaboration? And that, I'm assuming, gives you a sense of, okay, this is how we should design our tool.
This is usually how it starts. So if I'm at conferences and I present our work, people tend to ask me if it is possible to develop a probe towards their protein of interest or their target. I usually have a look, then also like how many synthetic steps that would be, like what our capacities are, and to foster a strong collaboration. I also look, of course, if like these proteins are expressed in beta cells or in neurons, so we'll have some overlap of our research interests. Yeah, but with 40,000 proteins, right, or 40,000 different proteins, I'm always very eager and happy to hear like what some things are doing I've never heard of before, in which context and so on. Yeah, but that's a great starting point usually to talk to people and not only go to ChemBio conferences, but also to Synapse conferences or molecular biology conferences and workshops alike.
Speaking of research interest, what are some of the research interests that your lab and your team has?
We are a very diverse lab, consisting of chemists that do synthesis, but also theoretical chemistry. We have biochemists, biologists, toxicologists. But all like under the realm of cell surface proteins and their communication in between. We do a lot of synthesis of new fluorophores that are good for imaging or better for imaging. This is where we start. We're looking for probes, and we're looking also like a little bit more into the biology now. So if I would have thought like that we explant pancreatic islets here a couple of years ago, I wouldn't believe myself, but we can do this also thanks to David, who was teaching us. But yeah, it's a very interdisciplinary group. What is important for me is that there's always overlaps and people talk to each other, right? That the synthetic chemists like us, like, hey, what is your roadblock or your bottleneck right now? Like, can we go there? Or like the biologist would say, like, hey, I need, this is what I would need. And it's all going around the GPCR or the cell surface protein things. So this is what we are doing.
With such a diverse group, which I think is quite unique, especially in a group where you have chemistry and theoretical chemistry and biology and all of these disciplines, how hard was it to develop that common language so that the chemists understand the biologists? And because it's difficult. We all speak different languages. We all have a different sense of what's important to us from a work perspective. And so I know the challenge is there, and I want to hear a little bit more about how you, as a team, overcome those.
Yeah. I think the crucial aspect is open communication and not being afraid to ask a question. Right? I mean, a question from a chemist to a biologist can sound absolutely idiotic, and vice versa. But just be allowed to ask those in group meetings. And this is like, okay, like I did not get that. Can you put this in other terms? And then like slowly approaching the issue at hand. And I try to engage a little bit as a translator. I'm also not a biologist, but I've seen a little bit more. So this is where we usually like come to a point that we all agree on and that we all understand.
That's wonderful. And speaking of, you just mentioned you're not a biologist. I'd be curious to hear about over the years in the context of your collaboration with David. I remember you mentioning you were a PhD student where you started, synthesizing molecules for him. How much did you learn from each other, and how did that communication between the two of you from a scientific perspective has evolved over the years?
How did it evolve?
Yes, what did you learn and what did he learn? Actually, I forgot to ask him, what did he learn?
Well, David certainly learned about the use of acetonitrile for conjugation reactions, as well as always bragging about. And I've learned about like the heterogeneity and the complexity of tissue, right? I was always working in 2D cultures to a point. And then, having this new realm and seeing so many challenges, which I'm excited to tackle, that was quite new for me.
Any aha moments that shaped your trajectory as a scientist over the years?
Generally speaking.
Yes, generally speaking. I think the tissue culture and the cells, the live cell aspect sounded like one of them.
Yeah, that was one of them. I think from a chemical point of view, I would say like we have a project running where we use isotope effects in chemical biology, which I'm quite excited about. Where it's like, wow, you learn of that during your studies. Yeah, there's a little bit of an isotope effect, and you use this for mechanistic studies. But after all, like it's not that big. But it is actually and we can use this to make dyes brighter or shaping the switching kinetics of small molecules and so on. Aha moments were obviously like the first use of in vivo of our probes. This was really where I was thinking, okay, this is the holy grail in chemical biology, right? Like you want to see this in the living and intact animal. And that was an amazing collaboration with Amelia Linnemann from University of Indiana, who did this with 2-photon microscopy, and like you can actually like see the islet lighting up in green in the mouse. And this is like when I saw this data, I was like, wow, that's amazing! Like this is opening so many new doors. And I don't know which door I first can step through, but still a lot of the list.
Wonderful. If you could dream big, what tools would you love to develop next?
Okay, generally, if I would dream big, I would still like to play the Champions League finals. So if anyone from Real Madrid is listening, I'm still available. But which tools I love to develop? I think what would be absolutely groundbreaking is to have a general approach for chemical tools. And this is actually something we are looking into, also driven by the latest developments with neural networks and artificial intelligence. They have one approach to say, like, okay, we can now deduce a molecule for each target, and ideally also in their respective pool. That would be a dream, that you can predict this, make it, and then light up the tissue in as many colors as you can.
I think, as we started talking, I was exactly thinking about this, about being able to have this whole entire catalog of pick your receptor, pick your probe, pick your application, and being able to do it, in cells, but also in tissue, in order to follow the activity and the localization and where the fate of the receptor in its native environment.
Exactly. And maybe not only one, but maybe 2 or 3 or 4, right? Like, because crosstalk and interplay, this becomes more and more acknowledged and important. So that will be absolutely amazing. Of course, with a nanometer resolution, I have to say.
Of course, yes. We
to make things easy.
And to your point, I think to your point, it's very important to address, not one receptor, but multiple receptors. And I think, when I was a graduate student, there was this whole debacle around the importance of dimers and higher level oligomers. And I think having the tools to visualize the receptors in tissues and living animals, being able to say, well, for this process, we need a dimer, or for this process, we just need a one subunit, or we need a heterodimer, or we have 4 receptors together doing other things, could be really, an absolute fantastic breakthrough.
Absolutely.
Any advice for young scientists or young chemists who want to enter the chemical biology / GPCR biology field?
Be curious, ask questions. That's my advice. If you're at a conference, engage with people, no matter like their position, their age, their maybe Nobel Awards or whatever, right? No, we all love to talk science, right? We all like to discuss problems, issues. And sometimes issues are easily resolved if you ask the right person, right? Sometimes problems just materialize when you start speaking about it. So this, like, follow your curiosity, ask questions, don't feel dumb or anything if this question is easily answered. You didn't know, a million other people also didn't know. So just go for it. And look what excites you technique-wise. I think this is very important. Look what excites you people-wise. Like, if you look for a lab, for instance, if you're looking for a PhD, for postdoc, that like resonates with what you are like. So go there, like, have a look, spend a day with them. Look what city is interesting to you, right? If you like a larger city, if you like a smaller city, if you like the environment, if you need the mountains to hike, or you need flat ground to drive your racing bike. These are important considerations, especially for a PhD, when you know it's going to be the next 4 years, doing a lot of labor, a lot of frustration, but also a lot of benefits, and some good feelings when the experiments finally hit the fan.
Yes, no, I think that's absolutely fantastic advice. When you were, let's say, a PhD student and a postdoc, what were some of the techniques that you used in order to, go to conferences and be able to talk to those advisors, to those potential Nobel laureates, to go out of that comfort zone?
Can you phrase this again? What was the?
What were some of the were you at all? Let me just give you an example.
Oh.
Every time I would go to a conference, and even now, sometimes I would feel like, I don't want to bother that person. I'll just email them and we can have a separate talk. And I had to go out of my comfort zone and at least go and introduce myself and say, loved your talk. This is, I have a question, and have a conversation with that person. But that takes that little extra push to go outside of the comfort zone. And I'd be curious, when you were a PhD student or a postdoc and you were at conferences, what were some of the things you had in mind when you went up to people who, for example, presented, to break that ice?
I think that there was also a good portion of naivety involved.
Which is good sometimes.
Yeah, exactly, which should be embraced. But yeah, I mean, you know why you may want to talk to that person, right? And then you say, the thing I did was like, hi, I am Johannes Broichhagen. I'm JB. I work for XY. I do my PhD. I'm running into a problem, and I know you're working on a similar field. Can I pick your brain for 1 minute? And it's always been more than 1 minute later, right? Because as I said, like people love talking about like their research, their ideas, and also tap into other things, right? We look at science as riddles. We want to solve this and go back and forth. And once the ice is broken, then usually everything's flowing.
Thank you for that very tactical, very specific advice. I think that's going to be absolutely valuable to our audience who needs to get out of that comfort zone. And it's as simple as saying, yes, can I pick your brain? I'm so-and-so, and I admire your work.
Right.
All right, last but not least, what impact do you hope your work will have on the GPCR research community? Other than the catalog of compounds, that's very specifically allow you to look at receptor trafficking in vivo.
Yeah, I hope the impact would be as broad as possible, obviously, not for personal reasons, but like to just like acquire more knowledge and to make things easier, right? And I'm a tool developer. I'm not a biologist. I like when things do work the first time. That's an important impact because otherwise it slowly but surely lands in the freezer and will never be used again. And then that people with their own research programs, with their own research questions, can actually like use this and see the benefit of it to be implemented in their lab.
Thank you for that, JB. I think that's very important. And then I have one last question. When it comes to cross-functional collaboration, what are some of the principles that guide you when you start developing relationships with potential collaborators? I know the answer to this, but I want to hear on your end, from your perspective, what's important.
I think two things are important: trust and fun. I think this somewhat goes hand in hand. If you trust your collaborator, that's not a one-way road. That's mutual, right? To say, okay, yeah, we want to like go forward with this, and we alike, and okay, there might be some things that are slow or whatever, but like we're working on it, or like we'll come visiting and see how we can solve issues. But knowing that you're both pulling on the same string. And then, of course, without fun or the evening pub, I think things would just be a little bit too, dry, like literally and figuratively.
No, absolutely, and I think you make a great point. Trust is important, and it goes both ways. And fun in the quest to finding the answers to the common questions that you're posing, you're putting out there. Wonderful. Thank you so much. I really appreciate your time. I had a really great time chatting with you.
Likewise. Thank you so much for the invite. It was a lot of fun.
Thank you so much.
Thank you, JB, for giving us a look at the chemistry and tool design driving modern GPCR imaging. If this conversation brought new ideas to your project or your team, share it with a colleague. It helps the community grow. Make sure you're subscribed to the DrGPCR podcast so you don't miss part 3 of this episode series or any other new DrGPCR podcast episode. And if you're listening on Apple Podcasts or Spotify, leave us a quick review to help more GPCR scientists discovery teams find the podcast. Today, you heard how better tools reveal receptor behavior. Terry's Pharmacology Corner complements that by strengthening how teams reason through what those receptors are actually doing. Dr. Terry Kanakin turns GPCR pharmacology into decision-ready frameworks for discovery programs. Link in the show notes to check out Terry's Corner. A huge thank you to Celtarys Research, our strategic partner, for supporting the advancement of fluorescent ligand technologies across the GPCR field. And as always, thank you to the DrGPCR team for making this episode possible. All the links are in the show notes, so go check them out now. And because receptors don't work alone, and neither should you. Until next time, stay safe.